Our next speaker is Donna Blackmond, who's from the Scripps Research Institute in La Jolla, California. Donna will be talking, Dr. Blackmond will be talking to us about autocatalytic models for symmetry breaking and the emergence of homochirality.
Thank you very much for the invitation to talk at this symposium. It's been great what I've seen so far, and really happy to share some of the work that we've been doing that I'm going to talk about. Actually, that's been going on for quite some time, and it has to do with chirality. We heard a lot about monomers, building blocks for origin of life. We've heard about some of the informational polymers that they make. The one thing that has sort of been left out a little bit so far is the fact that these are all chiral molecules that are all single-handed. The proteins are all left-handed, the sugars are all right-handed, and it's the single-handedness of these biological molecules that is seen as a fundamental signature of life. And so a lot of our work has been trying to understand how this came about. Because if you think about this, if it's a signature of life, if you go back to the early prebiotic soup when the first carbon-carbon bonds were being formed, that could go left or go right, they didn't really have any bias to go one way or the other. So the question really is, how did we get here, modern biology, where we are today, from there, which would have been a racemic prebiotic soup with an equal mixture of each end of the molecules. And so this is a question we've been talking about for a couple decades, actually, trying to think about. And it has really two parts. First, you have to break symmetry. You have to take that equal mixture and find some way to make an unequal mixture in a way that isn't trivial. Like every other bond that you make, every time you have an uneven number of molecules, obviously you have broken symmetry, but in a way that can be propagated. And that's the second part, is the amplification or the propagation of that broken symmetry. Because symmetry breaking doesn't get you where we are today. We have to find a way to get to, virtually all of one hand and not the other in biological processes. So, as I said, we've been working on this for a while, and the type of models that we've come up with have a range from purely chemical reaction models all the way to purely physical processes, and then in between models that sort of combine both physical and chemical processes. But I'm going to talk today about just this first type of model of autocatalysis. And it's actually the first work that we did, and it's some of the most recent. And so I've highlighted in red a ChemRev article that we wrote in this issue that's all about chemical evolution. And it kind of summarizes all of our work and other people's work in this area of autocatalysis, asymmetric autocatalysis, for the past few decades. So this really goes back a lot further back than that, and that is back to an article that was written in 1953, which was a good year for origin of life type papers. We just heard about Miller-Urey, and obviously that was also the structure of DNA. But this was an article by Charles Frank, and he was a crystallographer at Bristol, at Oxford, and then at Bristol. And he wrote a purely theoretical paper, mathematical paper, about what he called absolute asymmetric synthesis. And what he wrote is that if you, his mathematical model in words says that if you have a substance that can be a catalyst to produce itself, and can act as an anti-catalyst to somehow suppress, at least partially suppress its other hand, mathematically that's enough to get you to homochirality starting from a small imbalance. And he showed this with math and with graphs, no experiments at all. But the last sentence of the paper was what got a lot of people intrigued. He said, a laboratory demonstration may not be impossible. And so years later, Hans Winberg, who was a scientist in Groningen who worked a lot in this area, he said, no, that sentence, that's a challenge to any red-blooded chemist, a synthetic chemist. The theoretician is going to tell you that maybe you could find a reaction that does what he said. And so this was something that people worked on. It was really 40 years after that paper by Frank that challenge was met. And it was met by Kenzo in a paper in Nature. It's one of the papers that the Nature editors have called one of their top 50 papers of the last 50 years at the turn of the 21st century. And the reaction is it's not prebiotically relevant. It's not going to happen in an aqueous prebiotic soup, but it's an alkylation of a pyrimidal aldehyde with diisopropyl zinc. And it turns out that the product of the reaction can catalyze it, making it go faster, but also increasing the enantiomeric excess. Basically, you can start with adding a little bit of the product at very low EE, and you can get much higher EE out at the end of the reaction, which was the first example that seemed to demonstrate the Frank model. And I have to say that it's actually the only example still to date that we have found that has really been demonstrated to follow this model. And it's something that when we looked at this, we kind of thought of this as, we called it the ultimate nonlinear effect. If you work in catalysis or in asymmetric catalysis, you may know that in the 1980s, Henri Kagan published papers with something he called nonlinear effects in asymmetric catalysis. And it turns out that if you, the pointer here, typically when you do an asymmetric catalytic reaction, you use 100% EE catalyst. But if you can't make that, or for some reason you don't have an anti-pure catalyst, you would expect that each hand of the catalyst would do its own work and you'd be on the linear line. But it turns out in some cases you could get behavior that wasn't linear. And in particular, what was interesting and intriguing was positive asymmetric amplification. You get a higher EE in the catalytic product than what you paid for in the catalyst that you made. And so Kagan developed models to describe this, and it's become really a sort of a diagnostic now for catalytic behavior to try to understand the structure of asymmetric catalysts, not in an origin of life sense, but just in general. But we thought the SOI reaction might fit the Kagan type model for nonlinear effects in order to have this amplification of EE. So we decided to study it. We actually had been doing work in this area of nonlinear effects for a while, both in asymmetric synthesis, where you have a chiral auxiliary that does its job once, one and done, or catalysis or kinetic resolution, where you do turnovers, the same catalyst does the same thing every time. And then now asymmetric autocatalysis, where you make more of yourself, and each one of those can do its work over and over again. So it was kind of a nice segue for us to look at this after we'd been looking at these other areas of nonlinear effects. In each case, what we had to do is to develop a little bit of a modification of the Kagan models for catalysis, first to talk about asymmetric synthesis, and now to talk about asymmetric autocatalysis. So the Kagan model can be described pretty simply here. Basically, you make dimers, or he calls it ML2 model because many of the catalysts there were metal catalysts with two ligands. But essentially, you have two chiral species in the catalytic species. And so you can have homochiral species, SS or RR, and you can have a heterochiral species. And his model basically describes the equilibrium distribution between these species and the relative rate of the heterochiral species as a catalyst. The homochiral species would have to have the same rate. They would give opposite hands of the product, but the heterochiral species it's a diastereomer, it can have any other rate. And so the model really just has those two parameters, K and G. And so we decided to take a look at this and fit the system to the Kagan model, which we had to modify a little bit because after every time we make a molecule, every turnover, we've made another catalyst, and it may be left and it may be right. So the model has to, after each step, adjust for how much catalyst and how much left and right hand catalyst. Product equals catalyst. And so the first three runs that we did, that we fit to this model, was we took an enantiopure run, a racemic run where the catalyst was racemic, and one where the catalyst that we added was in between. And we fit it to this model. We asked the model to pick the best values of K and G, and it came up with a very good fit. We thought was quite reasonable fit. And it turns out what the fit said was that K equals 4 and G equals 0, which are actually pretty important parameters. K equals 4 means a stochastic distribution of dimers, which means that the system didn't have any preference. If you make an R and you see an R, you want to be a dimer. So if you see an R first, you become RR. If you see an S first, you become RS, which is actually an important feature if you're thinking about the prebiotic world where we didn't have the chance to bias reactions the way we do now when we develop asymmetric catalysts. So K equals 4 is stochastic distribution of dimers. G equals 0 means that the heterochiral dimer is inactive, that this reaction doesn't happen. And it basically could be slightly suppressed according to the Frank model, but here in this case, it seems to be totally suppressed. So this does seem to fit the Frank model quite well. We were also able then to take that model and apply it to other data sets. Here's two more runs. with just using k equals 4, g equals 0. And now I'm plotting, before I was plotting rate on the y-axis. Now I'm plotting fraction conversion versus time. Before you saw the autocatalysis as a peak, and now you see it as this sigmoidal shape. Here's a 22% and 6% EE run. And the experimental data in circles, and then the kinetic model on the lines. Because in this system we actually are producing, the product is the catalyst, we also can predict, the model predicts what the enantiomeric excess will be over time, how it will amplify from 6 or 22% up. So that these pink lines tell you what's predicted, and then we could just take samples, completely orthogonal technique to to the reaction calorimetry that we used for the rate data, and we're able to, and this was what the data said. Those are not lines through fit through the experimental data points. That's a prediction of those data points. So we felt pretty good that the model was really able to predict, solely from the kinetic data, we were able to predict the chiral amplification. And we've backed this up with, in the time since the model, with NMR spectroscopy of the relative, the k equals 4 was confirmed by NMR spectroscopy of the diastereomeric forms, and many other experiments as well. And every single run we've ever done with this reaction has fit this model. So that's quite a while ago, but that's just a setup for what we wanted to do next. And in the meantime, also, Kenzo Soi was going on and doing a lot of work in this reaction. He was continuing to show how the ways this reaction works. We know that the product itself catalyzes the reaction, but he also showed that you could catalyze this reaction with almost any chiral source that you could imagine. You could catalyze it with chiral alcohols, chiral minerals like quartz. Quartz has 2 enantiomorphs. And if you threw one of those enantiomorphs into a Soi reaction, the product would go one way. If you threw the other enantiomorph in, it would go the other way. You could shine circally polarized light on a racemic mixture of secondary butanol, and then take that mixture and use it to catalyze the reaction. The circularly polarized light degrades one hand of the enantiomer a little faster than the other. And he showed that with fidelity, that all these things could happen. But the most interesting or intriguing thing to me was something that Soi showed about a little more than 10 years ago. And he showed that you could direct this reaction faithfully in one direction or another with molecules that were chiral only by virtue of an isotope. The one I'm showing here is what we worked with, but which is an OCD3 group versus an OCH3 group. Otherwise, that's a completely symmetrical molecule. He showed it for C12, C13. He did it for nitrogen isotopes, oxygen, and he also looked at it for some HD. And again, it seems like such a tiny, tiny difference that could nudge this reaction faithfully one direction or the other. So we wanted to try to understand how that happened. And so we started to look at this reaction that's at the bottom here. And it has a little bit different shape. If you remember, we saw the sigmoidal shape in our early work, but it happened much faster. And it didn't have this long sort of lag time in most of our cases. So this was interesting. And then it just seems to light off after a while. So we did a lot of work. I'm not going to talk about most of this paper because I want to get to what we did next. But basically, you can read the paper and hopefully it convinces you what I'm saying here, is that what we found is that the initiator is actually an inhibitor. It inhibits the reaction in the beginning. If you don't put any product or anything in this reaction, it will go. It will go down the middle. It will make stochastically racemic mixtures every time you do it, because the background reaction can be quite high without the catalyst. If you have the product in there, the product reaction is much, much faster. But what was happening in this case, we didn't put any of the reaction product in to start with, just this initiator. And so what happened is we think from the data that we've shown by both proton and deuteron NMR, and by doing diffusion measurements in NMR and also DFT calculations, that we make a 2-to-1 product initiator complex, 2 products for every initiator. And this binds quite tightly. So the autocatalysis doesn't have a chance to start because we can't make our dimers that we want to make from our product because they're tied up in the initiator. But as soon as the initiator is all tied up, and then the next products that are formed in the background reaction can go on and trigger autocatalysis. And so that's how we think the initiator is working. And so basically the first products that are made after this initiator complex formation is done are what are responsible for the autocatalysis. And they also are responsible for the amplification. It's not going down the middle. It is going one direction or the other. And so our model for what we think is happening is that when the product binds with this isotopically chiral initiator, it makes a complex that have slight differences in energy. We couldn't actually pinpoint how that energy difference by our DFT calculations. I think it's even too small to see by those methods. But this very slight difference, which we're going to talk about as gamma, the difference between the major and minor complexes, is enough to result in a very tiny difference between the amount of RNS that's available at the point when the initiator has done its work and the autocatalysis is able to take off. And if R is a little bit more than S in this case, then essentially R would go on and it would win out over, it wouldn't completely block S in the beginning, but since there's more of it and it's exponential, it wins in the end. And that's what we think is happening in this case. So this is a really tiny difference that we're talking about. And so this led us to think that maybe we could use this reaction of the isotopically chiral initiators to help understand symmetry breaking. So far, we've been interested in looking at the amplification part of the picture. How do we amplify a small imbalance? But now let's think about how did we, this model is telling us how we got this imbalance. And can we use that to try to figure out, how small is that? What kind of energy do we need to make the reaction not be stochastically going sometimes left, sometimes right, but always going one direction? And can we quantify that energy difference? And so before we talk about answering that question, we want to go back and think about fundamental forces. Okay? There are 4 fundamental forces: gravity, electromagnetism, the strong force, and the weak force. The first 3 exhibit what the physicists call parity inversion. The weak force does not exhibit parity inversion. And parity inversion, you can think of it as the way physicists talk about chirality. It's an operation that converts a physical phenomenon into its mirror image. And so basically everybody else is symmetric except for the weak force. The weak force is asymmetric. Okay? This was first found out theoretically in the 1950s. The weak force, what they say is that it violates parity. And the theory was developed by 2 Chinese-American scientists in 1956, Li and Yang. Okay, and it was just within a few months after that, the whole community got excited about it. And Xin-Cheng Wu, a very meticulous female scientist at Columbia, I think, within a few months, she had demonstrated experimentally that this is true. And that was really important because nobody believed it when Li and Yang first came up with the theory. Wolfgang Pauli and Richard Feynman both bet against it being true. But by the time Xin-Cheng Wu and then other people also did experiments on this, Dick Garvin and others, a number of experiments happened in quick succession that showed that it was true. And so, often when people win Nobel Prizes, it takes like 10 years after they do the work. Well, this work won the Nobel Prize in 1957, and they gave it to Li and Yang, even though there's a rule of 3, right? They did not give the prize to Xin-Cheng Wu, even though nobody believed it until she did her experiment. So anyway, that was a long time ago, and I think people realized that they had kind of left her out because she was the first year that the Wolf Prize came about, she was given the inaugural Wolf Prize. So, a little background, and so back to our question about, well, first, what are the implications of this, of what these guys found? So the implications are huge, and I think that's why when people realized it won the Nobel Prize so quickly. If the weak force wasn't asymmetric, we would not be here. There would be no matter. Matter and antimatter would essentially annihilate each other. So we have matter because of the weak force. But then this led to, about 10 years later, the corollary or an implication of that was published by Yamagata, and he said, wait, if that asymmetry in the weak force is true, that means that what we tell our students in class is not true. We tell them that enantiomeric molecules, the left and right hand, are absolutely identical with all their properties. But this turns out that because the weak force is asymmetric, there's a tiny energy difference between the left and right hand of a molecule. Okay? And that's what's called parity violation energy difference. And so some people, Yamagata's put forth this proposal that maybe this minute difference could actually be enough to rationalize biological homochirality. That was another bifurcation, like matter-antimatter. The number has never been measured experimentally. People like Martin Quack, who I quoted on the last slide, is trying to measure this, but it's been calculated, and it's a lot of calculations have been done. It's a very, very small number, as you can see. So we thought about our experiment here. When we had used just the enantiopure initiator, we got this difference between the two. But if we had used both R and S initiators, you would think you would get back to a situation where there'd be no difference, no bias, you'd be going both ways. Okay, so what if we take our initiator and we just lower the EE value slowly and look at what happens until we get, when do we lose the fidelity going one way or the other? At that point, where we get stochastic behavior, we can actually calculate the energy that's required. We can basically calculate this gamma value, and we can calculate this alpha value, which is what we're going to need to understand to break symmetry. So now we already have a great kinetic model of this system, so we're just going to have to add this stochastic idea to it. First, we wanted to figure out where this happened. So we basically took the initiators, left and right hands, and we just kept looking at what happened as we went down. And you can see that somewhere red up means R, blue down means S, and sideways means it stayed racemic. So you can see we're starting to lose the definition, somewhere between 1 and 0.1% EE. I'm not going to be too bothered about the exact numbers. I just want to try to use that to get like a threshold energy. And we do already have a kinetic model that we understand the SOI reaction quite well. And what we wanted to do here was to just add to each elementary step in the model, add stochastic noise, and try to figure out the value of gamma that gives us, directional behavior, that we lose it at 1% EE and we still don't lose it at 0.1% EE. Basically, we get low enough that we stay stochastic at 0.1 and we get directional behavior at 1. Okay, and so this is what we look at. At 0.1% EE, for all these values of gamma, we're still going both directions. It's stochastic. But when we look at 1% EE, when we get down low enough, gamma is 2.6 times 10 to the minus 6, we start going faithfully in one direction. And that alpha critical is this value that's here in the boxes that says if I go past that point, it's no return. I can start going one direction like this guy, but then I turned around and came back. But here, if you get to alpha crit, you're never turning around. That's the point of no return. So now our model has these two values. And basically, what can we do with it? Well, from that alpha crit, we can calculate the EE that we need. If we were going to put the chiral initiator in, if we're going to put the product in this reaction, what EE of product would we need, how low before we would lose and go into stochastic? It turns out it's really low. 3.5 times 10 to the minus 7 to 10 to the minus 8 EE, that range. So I tried to measure this experimentally. He tried to go down, 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 and he lost it at about 10 to the minus 50 EE. He couldn't get reproducible results after that. You can see that's still 2 orders of magnitude higher than the EE that will still give you a bias in one direction. So it's an incredibly sensitive chiral reaction. Another point about the SOI reaction I just want to make is how robust it is. If this were a typical catalyst, and we started with a 10 to the minus 7% EE, basically, if you think about a catalyst, how many turnovers it does, if we just had one molecule doing its turnovers, that would be 10 billion turnovers. It's hard to talk about turnovers in autocatalysis where you're making more catalysts, but it takes a lot of reactions to get the homochirality when you start with such a low number. So that's important, that the robustness. The other question is about this alpha critical. That's a very small molarity, right? 5.8 times 10 to the 11. So we tried to think about a way to describe that, how low that is. And what it is if I take 20 milligrams of the autocatalyst and I throw it in an Olympic-sized swimming pool, that's the concentration of 5.8 times 10 to the minus 11 molar. So it's extremely sensitive, this reaction. It's rather mind-blowing how sensitive it is. So basically, we can calculate the energy that from this EE value, just the same way that you could look at delta delta G's for any asymmetric reaction. And it turns out that it's about 2.8 times 10 to 2 times 10 to the minus 8 kilojoules per mole. And if you compare that, remember we talked about parity violation energy difference. That's 10 to the 12th to 10 to the minus 10 to the minus 12th to 10 to the minus 15th. So we're still orders of magnitude away. which would suggest that parity violation energy difference wouldn't be enough to tip us one way or the other. It certainly isn't enough in our chiral world today to make any difference to any of the reactions that we're doing. But there are people who have models to show how you could still pull signal out of noise if you have enough time and enough small values of alpha. So I think that I'm just going to go straight to the conclusions now. This probably PVD is unlikely to be sufficient to count for symmetry breaking, at least in the SOI reaction that we studied. And you're going to need a reaction like the SOI reaction, but very persistent and very efficient if it's going to reach homochirality. And so far, the SOI reaction is the only one we have. It's maybe more likely that we have to think of some combination of processes, not just a single autocatalytic reaction that would get us to homochirality. So that, and along those lines, a lot of people, our group included, are trying to find more prebiotically relevant reactions that could do what the SOI reaction does. So far, we haven't been able to find one. So that's a kind of a holy grail for anybody that's interested in thinking about what kind of reaction that would be prebiotically relevant where the product could then make itself and amplify EE. Okay, so that's it. I just want to acknowledge we aren't part of the center here, but we've been very lucky to be part of the Simons Foundation collaboration on the origins of life, which has been going on since 2013. And it's just been a wonderful, amazing group of people, all the way from astrophysicists to astrobiologists to geochemists to prebiotic chemists, RNA world people. And it's been a great fun working with them. And all the work on the chiral initiators that I talked about was done by Neil Halbecker, a student in my group. And we worked a little bit with Dilip Kondaputi, who helped us with the modeling because he's done a lot of stochastic modeling. So thank you for your time.